I want to wish everybody a happy Halloween. Several weeks ago, I saw a news article that was talking about some exciting new molecule called TTK21. And the article was referring about a small molecule that had the potential to help spinal cord injury, to help neurons regenerate neuronal pathways. And so let's go ahead and talk about this molecule, TTK21. There was a study that showed that small molecule could stimulate axon growth in mice that had severe spinal cord injuries. You can see a micrograph here where they're trying to show in these green spots here new axonal growth to develop connections between nerve cells. And the paper says that it's a small molecule epigenetic activator called TTK21. And let's go ahead and take a look at the biology, maybe just briefly comment on that, and then the chemistry behind TTK21. And what really caught my attention was the fact that TTK21 looks very much like the kind of molecule that you should know how to synthesize from taking a class like this. It says that TTK21 is a weak activator of cyclic AMP response element binding protein and related to P300 acetyl transferases. That to me sounds like a histone. That means epigenetics, the way DNA coils around these histone proteins and then is unraveled for gene expression. But when you look at the chemistry behind the synthesis of TTK21, it's all about making ethers, making amides, and that's stuff you know how to do. You already learned in the first quarter of organic chemistry how to make ethers. You've learned already how to make amides and do functional group interconversions with carboxylic acid derivatives. So let's go ahead and take a look at the chemistry behind this. The key thing that they're trying to do here, starting off, is to make an ether bond. And you learned about this process called the Williamson ether synthesis back in your first quarter of organic chemistry. And you may have actually made an aryl ether in the Chemistry 51 laboratories that go along with this course. What they're doing is they're taking this very common starting material, acetyl salicylic acid, salicylic acid. That's what they use to make aspirin by acetylating that. They treat that with a base, unfortunately an insoluble base, so they probably had to wait quite a while for things to dissolve. And they were deprotonating this phenolic group to make the phenoxide anion so that they could do an SN2 reaction and make an ether bond. Now, potassium carbonate is a base, and that means the first thing it's going to do is deprotonate the most acidic group, and that's the carboxylic acid. That's going to be the first thing that gets deprotonated. So they can't stop it from forming this dianion, and they must have observed some SN2 reaction with the propyl iodide with this carboxylate, so they just went ahead and added 2 equivalents of propyl iodide. And you can see that the bond they're forming here, the ether bond, is very traditional. You take an alkoxide or a phenoxide, you do SN2, you make an ether. But they were also generating the ester through a non-traditional mechanism by doing an SN2. And that's not the normal way to make esters. The normal way to make esters is you attack carbonyls. They didn't even want that ester there. That was an accident. They didn't want it. And so they went ahead and did a saponification reaction. That's the last reaction we taught you about in our previous lecture on Friday. We taught you how to do saponification with sodium hydroxide. And then you add the proton back. After using saponification, you have to reprotonate the carboxylate using an HCl workup. So they got rid of that ester that they didn't want, and that sets them up to now make an amide. And the way they made an amide is exactly through the reaction conditions that we taught you about. So they use thionyl chloride and heat to convert carboxylic acids into acyl chlorides, into acid chlorides. And now you're ready to make an amide bond in 100% yield. So they take their amine that they're interested in this case it's an aniline, it's an aromatic amine, and they use 1 equivalent of triethylamine as a base to soak up the HCl. So just like we taught you, they probably sat in a class like this and learned chemical transformations in a class like this, and now they're making potential cures to spinal cord injury, and that's pretty exciting. So I hope you appreciate the amazing chemistry that we're learning in this class. It's actually useful chemistry for curing diseases and impacting human health. When we left off on Friday, we almost finished chapter 20, and this is the last part of the material for your second exam that's going to be coming up in less than 2 weeks. So when we left off on Friday, we had talked about saponification, how to use sodium hydroxide, not heat, just sodium hydroxide, room temperature, to cleave esters efficiently to make sodium carboxylates. And then you can protonate those using an HCl workup to make carboxylic acids. Now we're going to talk about a much tougher problem, and that is how do you cleave amides? You know, nature didn't make your proteins using ester bonds. You know, in neutral water, esters have a half-life of like 7 days. Amides have a half-life of 7 years in neutral water. So you can see why nature made your proteins using amide bonds. If you're in a laboratory and you don't have an enzyme that cleaves the amide bond, it takes really harsh conditions to efficiently cleave amides. And so let's go ahead and talk about those two types of harsh conditions for cleaving amides in order to get carboxylic acids and amines back out. So the first set of conditions I'm going to tell you about involves using a powerful, not a powerful nucleophile, but powerfully nucleophilic conditions. And what you do is use sodium hydroxide. Sodium hydroxide can add to carbonyls, not quickly. And what is it that makes these conditions powerful? It's not the nucleophile, because hydroxide isn't that powerful. It's the fact that you use highly concentrated sodium hydroxide. Not 1 molar, not 2 molar, not 3, 4 molar sodium hydroxide. You do those reactions in water. And the other thing that makes these conditions powerfully nucleophilic is you are heating the heck out of this. You're boiling it. That's the highest temperature you can get to in an unsealed system is 100 degrees. And you boil it for hours and hours and hours. And that's what it takes to cleave a bond like this amide bond. Not easy to cleave. Now the product of that reaction is not a carboxylic acid. The product of that reaction is a sodium carboxylate salt, just like saponification. So if you want the carboxylic acid so that you can make acid chlorides or something like that, you have to do a workup with that. You have to do an HCl workup. You can't protonate the carboxylate anion using water. You have to use HCl. And so that would generate the carboxylic acid. And if that's what you need to do to neutralize it, if you want to extract that into an organic solvent and isolate the pure carboxylic acid. Now the other product of this reaction doesn't need any sort of treatment to make it neutral. The other product is the neutral amine. That's this thing down here. So if you're trying to get your hands on the amine, you don't need any special workup. You just extract the amine directly into an organic solvent. And by the time we get to chapter 23, you're going to be all about those amines, making amines and using amines. Now what if you don't have a bottle of sodium hydroxide, right? What if you just wanted to use water as your nucleophile? That's not so easy. Water is not a good nucleophile. Sure, it can attack carbocations, but anything can attack a carbocation. If you want to get water to attack this carbonyl group, you have to use HCl, right? Hydrochloric acid will stick a proton here on the carbonyl. You'll generate small amounts of this protonated carbonyl, and that's what it takes to get water to attack an amide. But that's not enough. Amides are so resistant that you need to go beyond just the HCl in the water. Concentrate, you need high concentrations of HCl. And you have to reflux this, boil it in water for hours and hours and hours. Doesn't matter whether you're using base, whether you're using acid, you got to boil these reactions for hours and hours. So high concentrations of acid catalyst, water, and heat, and that's the formula, the second formula for cleaving amides to give you carboxylic acids. Now, you get, from those acidic conditions, you directly get the carboxylic acid. You don't need to protonate that using an HCl workup. If for some reason you wanted the neutral amine out of this reaction, then you'd have to treat that with sodium hydroxide, because the other product of this reaction is the chloride salt, the ammonium chloride salt, because you're stirring it around in hydrochloric acid. Of course, the amine is going to end up protonated. So if you wanted to isolate the neutral amine, you'd have to use sodium hydroxide here, NaOH, and that's what would give you this. That's what would give you the neutral amine if that's what you wanted under the acidic conditions. You'd have to work that up with sodium hydroxide. All right, there's a section on beta-lactams, and those are cyclic amines made of 4-membered rings. They are strained. If you've taken a biology or a pharmacology class that talks about drug classes, you may have heard of beta-lactam antibiotics. That's like penicillin is a beta-lactam antibiotics. It has this very strange square four-membered ring amide, and that's like a mousetrap. It is just ready to spring open and acylate enzymes that are responsible for bacterial cell wall biosynthesis. Powerful antibiotics still used clinically today and in prescribed medicines today. But I'm not going to ask you about that for my tests. I mean, if I asked you how to use these chemical conditions in a cyclic lactam, I expect you to know what the products would look like, but I won't ask you about beta-lactam antibiotics, but it's good reading. All right, the chapter finishes up by giving you a summary table of all the transformations that you need to know for our next exam, for exam 2. You need to know all the different transformations that you can do with an acyl chloride. Starting from converting acyl chlorides to amides, you need to know how to convert acyl chlorides into esters. You need to know how that acyl chlorides hydrolyze. Usually that's accidental. Somebody somewhere, I'm not going to ask you this, but it's possible to take acyl chlorides and convert them into anhydrides. I already told you that in practice, in most organic chemistry labs, we buy our anhydrides, and they're made in a different way. We don't make anhydrides from acyl chlorides. So this table also tells you things that you can't make in one step. You can't directly convert an ester into an acyl chloride, or you can't directly convert an ester into an anhydride. Who would want to do that? So everything here that has a check mark is covered in this book, in this chapter, for how to do that transformation, that one-stage transformation to make these different functional groups. Now, the end of the book has 2 additional sections, one on natural and synthetic fibers, like nylon is an example of an amide polymer. I'm not going to ask you about that. It's cool to read about it. And the book also talks about biological acylation reactions. So you may have heard of acyl acetyl-CoA when you had to memorize those stupid biochemistry cycles, like the citric acid cycle. You know, I'm not going to ask you about that, but this chapter, carboxylic acid derivatives, is so central to biochemistry, drug synthesis, modern chemistry. You really need to know all the transformations that we've talked about in this chapter. But I'm not going to ask you about the biological acylation reactions or nylon synthesis. That's just kind of fun reading. All right, that's the end of our material for our second exam. This is the end of chapter 20, and we need to reset ourselves and get ready for the exam material that's going to be for exam number 3. And so let's go ahead and head off into our next phase here of thinking about organic chemistry. So you may recall that on our first exam, it seemed like a lot of material, but that was just one chapter. All those reactions of aromatic compounds, doing electrophilic aromatic substitution, a little bit of nucleophilic aromatic substitution with benzynes and addition elimination, and then some side chain functional group transformations like hydrogenation of nitro groups. It seemed like a lot of material. That was only one chapter. Your next exam is going to be on addition to carbonyl groups. We're going to talk about that stuff from chapter 17 where we taught you how to add Grignard reagents to carbonyls and hydrido reagents to carbonyls. Super powerful, super important. Of course, I'm going to be putting that kind of stuff on the next exam. Also, cuprates were in that chapter. That has some utility as well. Remember chapter 18 where there was all those transformations of aldehydes and ketones? Not esters, not amides, but aldehydes and ketones. And we told you how you could condense to make imines and enamines and acetals, how you could do Wittig reactions to make C double bond C out of a carbonyl. And then they threw in hydrocyanohydrin formation in that chapter. So you need to know those reactions in chapter 18 of aldehydes and carbonyls. Chapter 19 didn't have a lot of meat in there. We expect you to know pKa's of OH compounds and how acidic they are. In the end of the chapter, they talk about additions to nitriles, triple bonds. In nitriles, how you can add to those triple bonds in a similar way that you add nucleophiles to carbonyl groups. And this last chapter here that we just finished That's nucleophilic acyl substitution. Acyl chlorides, amides, esters, converting one back and forth into the other. And that's really fundamental. So our next exam is all about adding to carbonyls to do chemistry, acyl substitution reactions and additions. So now we're getting ready to start on exam 3 material, chapter 21, with a set of functional groups called enols and enolates. And let's go ahead and get started with that. Okay, so in this chapter, we're going to have 3 types of transformations that are important, and I'm going to try to generalize those transformations here. What we're going to show you is that if you take just a simple ketone or an ester, that you can add powerful bases. And obviously, I'm not willing to tell you about that. It's going to require some discussion here. But you can add powerful bases, and you can remove a proton from the carbon atom that is next to that carbonyl group. You can remove a proton to make a species that we call an enolate. Wow, that's a powerful transformation. You need powerful bases for that simple transformation. And once you make those carbanions by deprotonating, you're ready to start doing SN2 reactions with those. We can do SN2 reactions. Let's just suppose I put in ethyl iodide or ethyl bromide. I can use that carbanion that I just made to make a new carbon-carbon bond. So, right, see that bond right there? You can make that using an SN2 reaction. And you already know about SN2 reactions, you just don't know about this secret functional group called an enolate. That's this chapter. So chapter 21, we're going to learn how to deprotonate the carbon atom that is next to a carbonyl, and we're going to use that for SN2 reactions, because that's kind of, the book does that because that's all the book really seems to focus on SN2. Now, 50 years ago, we didn't have these powerful bases that I'm going to tell you about. So 50, 60 years ago, they had, just kind of traditional bases like carboxyl, like alkoxide anions, like T-butoxide and sodium ethoxide. Those aren't really powerful bases. They're bases, sure, but what they found is that if you have 2 carbonyl groups, 2 of them, now it's acidic enough for an alkoxide base to efficiently synthesize these carbanions. Carbanions between 2 carbonyl groups are so stable you can make them using alkoxide bases. So here you can use an alkoxide base. And once you make these types of carbanions, these enolates, you can't isolate these enolates. There's no way to isolate them. We're not going to isolate them. We're not going to talk about that. We isolate neutral compounds that we can extract into organic solvents. But if you make this enolate, you can do SN2 reactions with it, just like I showed you before. You could displace tosylates, you could displace bromides. And again, here it works because just an alkoxide base can deprotonate a CH if it's got 2 carbonyls as neighbors. And again, this is chapter 21 stuff. We'll talk more about that this chapter. You don't know enough yet to be applying this yet, but we're going to get to that. And you'd use that to make that bond right there. Now there's a third type of transformation that is in this chapter that it doesn't involve negatively charged enolates. We'll talk later about the mechanism for this other transformation. This other transformation involves taking ketones that have H's on the neighboring carbon and putting a leaving group there. And the leaving group that we'll teach you how to put on is a Br leaving group. And here's the conditions we'll teach you about when we get there. So I'm giving you a preview of the recipe, bromine and acetic acid. And that puts on a leaving group. And once you put on a bromine leaving group, well, now you can do SN2 chemistry. Now we don't use alkoxides for this SN2 chemistry because that's basic. And once you start throwing alkoxides in with this, it's not going to be SN2 city, it's going to be doing this kind of business. So, but you will see us use these alpha bromo ketones to add sulfur and nitrogen nucleophiles. So we'll be adding sulfur and nitrogen nucleophiles in order to displace bromine. So if you have a nucleophile here, we can make new bonds. You already know how to do SN2. I shouldn't have to teach you about that, but that's just traditional SN2 chemistry. And this is what you would do if your nucleophile is like a thiolate anion, or maybe an amine group, some sort of an amine. We'll have to talk more about those mechanisms. That's this chapter. So enolates for making carbon-carbon bonds, and then we're going to talk about putting a bromine leaving group alpha at the carbon atom that is next door to a carbonyl. Now, we need to talk about a special form of isomerism that was kind of hinted at in an earlier chapter. And that special form of isomerism is referred to as tautomerism. And instead of saying the word isomers, we have a special name for isomers that are different only in the position of a proton. So way back in chapter 11, that was the alkynes chapter, we taught you that if you take an alkyne and you do oxymercuration to add the elements of H2 and O across that alkyne, that you could make methyl ketones. And it might have seemed sort of mysterious at the time, right? This idea that we can make a methyl ketone out of an alkyne. There's a lot going on in that reaction in terms of bond formation. But what they mentioned to you and didn't discuss the mechanism of, or didn't relay the importance of, is that the reason you form this ketone is you're initially going through an isomeric compound. So the initial oxymercuration reaction, the mercury-catalyzed hydration reaction, is adding the elements of water across that double bond. We normally don't draw the H on that alkene. I just want you to see that we're adding water across this double bond. The elements of water end up attached here. And there is an unstoppable isomerization of this isomer here, referred to as the enol tautomer. There is an unstoppable isomerization to the keto tautomer. If I gave you a sample of enol tautomer, within 1 second, all 6.02 times 10 to the 23rd molecules would isomerize into the keto form, into this keto tautomer. You can't stop this isomerization. It's not stoppable. All the enols want to form carbonyl groups, C double bond O, right? That's the end point of organic chemistry. That drives organic chemistry. Global warming, CO2, everything wants to form carbon dioxide because it has 2 CO double bonds. So you can't stop this isomerization. And the general theme here is that carbonyl groups are more stable than enols. Keto forms are more stable than enols. For acetone or a methyl ketone, the equilibrium vastly favors the keto tautomer, right? We don't even talk about the enol tautomer. There's such a small percentage in there. So you don't see significant amounts of enol tautomer, even if they're there at equilibrium. We can't see them in the NMR. The amounts are too small. So keto forms are more stable than enol forms. But I'm going to give you an example here where, 2 examples where keto tautomers are less stable than the enol, that you want the enol. And the first example is going to be aromatic alcohols, phenols. You already know that the alcohol form is the favored form of that. We don't talk about the keto form of a phenol, but let me draw it for you just so we can put it up here for some discussion. Like the keto form of a phenol would look like this. There would be an H there, and you could start off with any substituent you want, like a bromine or a methyl group, and you can draw the keto tautomer, but that is massively unfavored, right? You very much want to prefer. So sure, carbonyl groups are stable, but obviously the hydroxy form, the enol form, is aromatic. And that's more important than the strength of that C double bond O in this case. So, phenols are one example of a stable enol tautomer. There's a second example, the only one we would show you or mention, of an enol tautomer that just happens to be stable. And if I draw the keto tautomer, it'll be more recognizable to you that this is simply 2 carbonyls that are kind of 3 atoms away from each other. We would refer to this as a 1,3 diketone, this compound over here called acetylacetone. And so why is it that the enol form should be favored? And it's not massively favored. It's only favored by about a 3-to-1 ratio. That's the equilibrium amount. right? It will immediately, if you take either one of those molecules and put it in an NMR tube, it will immediately equilibrate to give you this 3-to-1 mixture. If you look at the NMR, you see that 3-to-1 mixture. It's a mess. It doesn't look like one pure compound because you can't stop these molecules from equilibrating. You got 6.02 times 10 to the 23rd molecules in your NMR tube, and they're equilibrating back and forth, and you can't stop that. So why should the enol tautomer be favored in this case, even if it's only 3-to-1? Well, there's resonance. You get powerful resonance here where this enol can donate into that carbonyl group. That enol is the focal point for chemical reactivity right there, and it's nucleophilic. It's donating into the carbonyl, so that resonance is powerful. Helps to stabilize the enol form. And you don't have that up above here. You don't have that kind of resonance stabilization to stabilize the enol form. There's a second factor that stabilizes the enol form of this 1,3-diketone, and that is this hydrogen bond. There's a hydrogen bond here that stabilizes that form. I'll just write H bond. And that stabilizes that. Not completely, 3-to-1, but the lesson here is generally keto forms are more stable. Keto tautomers are more stable. You can't stop the equilibration, and there's just a couple of cases where you can actually see enol forms to be stable enough to look at using spectroscopy. So, but let's go ahead and talk about the acid-catalyzed tautomerization mechanism. So if you take any sort of a keto compound that has even one single H on this carbon atom. Now, again, we don't usually draw those H's, but they're the focal point of everything that's happening in the next 2 chapters. And this is why it's essential for you to know exactly how many H's are on every single carbon, especially the carbon that is right next door to the carbonyl carbon. So if you have any kind of acid, a whiff of acid, any acid present in the presence of a keto tautomer, you're going to catalyze this tautomerization. The surface of glass, of borosilicate glassware that we use contains OH groups. There's SiOH groups, and they're acidic. We use silica gel, SiOH, for chromatography. That means there's acidic groups on our silica gel. You can't get away from proton sources. If you have an alcohol, that could serve as an acid, not a powerful one. But this is such a facile tautomerization that you can't stop this equilibration between keto over here and enol. You can't stop the equilibration. So let's talk about the 2-step mechanism, the 2-step arrow pushing mechanism for that tautomerization, that isomerization that involves moving a proton from one place to another. So under acidic conditions, the mechanism involves first sticking a proton on the O, right? You've got to put a proton on there eventually. Under acidic conditions, that's step 1 of the arrow pushing mechanism. And so now we're going to generate this intermediate. It's a kind of species that you've seen before. You've seen these kinds of species before where you have a protonated carbonyl. That's an oxonium ion if there's 3 bonds to oxygen. But you still have that methyl group. And now I'm going to draw one of the 3 H's on that methyl group because I need that. You can see over here in the product there's only 2 H's. So I'm going to have to remove one of them, right? There's 2 H's on this methyl group or on this methylene group in the product, so I better get ready to remove one. So now I'm going to draw it because this is the last step of our 2nd-step mechanism. When we deprotonated our acid, we left over this conjugate base form of our acid, A-. I'm going to use that to pull off the proton. Which proton do you pull off? Well, half the time you pull off the proton from the oxygen, the other half the time you pull the proton off the carbon. Let's go ahead and draw that out. Pull the proton off the carbon and give the electrons to that super electrophilic protonated carbonyl. And that's the 2-step mechanism for this special kind of isomerization that we call tautomerization. So we call that tautomerization. Now I expect you to know this mechanism backwards and forwards. Let me go ahead and draw out the backwards mechanism for that. Suppose I give you an enol and it's in the presence of acid. Like the glassware, like anything. I can draw out an acid-catalyzed mechanism. Right over here when I was done I ended up with acid. If I just draw the same intermediates out, if it's truly an equilibration and it's going backwards and forwards, it has to go through the same intermediates. Equilibration processes always proceed through the same mechanistic intermediates. There can't be some secret pathway that the top molecules didn't know about. I'm just going to redraw the same intermediates with the same charges. What's different when you do the backwards mechanism is the arrow pushing. The intermediates will be exactly the same. The transition states will be exactly the same. Just the way we draw the arrow pushing will be different. So let me go ahead and draw out the backwards mechanism. So you can see here, if I'm going backwards, I need to stick a proton on this carbon right here. And I need to make a double bond here. So let me go ahead and do that. Okay, I've got these lone pairs on the oxygen. When I see an enol, this is what I'm thinking. When I see an enol, the first thing I want to do is take the lone pairs on that oxygen and push down into the alkene. That explains why the alkene of an enol is so nucleophilic on this yellow carbon here. This yellow carbon right here is so basic and nucleophilic. So it's the oxygen lone pair that makes that yellow carbon basic and nucleophilic. So I'm going to take these pi electrons. Don't start your arrow from the yellow carbon. That's not the way we do arrow pushing. When you do curved arrows, your curved arrow starts with the pair of electrons. It doesn't start from the atom. So we're going to protonate that pi system, and it protonates on the yellow carbon, and that gives us this oxonium intermediate. We still have this oxonium intermediate in here. And now the A minus that's in here can pull off a proton. Half the time it pulls off the CH and you go back. That's not interesting. The other half the time it pulls a proton off of the OH plus, and that regenerates our product, or whatever, the keto form. And it regenerates our acid catalyst. So this is the mechanism for acid-base tautomerization, going back and forth between enol forms and keto forms. I expect you to know this two-step mechanism. It's only 2 steps, and I expect you to know that because it teaches you about the nucleophilicity of enols on this carbon. Right? That's super important to know that enols are nucleophilic on that carbon. And a little hint here. There's a class of molecules called enamines that you learned about back in chapter 18. And enamines are even more nucleophilic on that yellow carbon. That's why people used to make enamines back in the 1960s and 1950s. So, okay, so you can also catalyze this tautomerization mechanism using basic conditions. Using base, so I didn't mean to cross that out, using basic conditions. But I promise you, if you start to make enolates, if you start to use base in the presence of carbonyl groups, you're going to get a whole nother set of chemical reactivity that is not, enols are not going to be an important actor or an important synthetic intermediate or mechanistic intermediate once you start throwing base in with carbonyls. That's our next chapter. I'll tell you what happens when you throw in bases with carbonyl compounds. And this is, enols are not in any way important under those conditions. All right, so what are we going to be doing this chapter? What are we up to? We're going to be taking protons off of carbons, ripping protons off of carbons using bases, and then we're going to be using those carbanions as nucleophiles. And you've already seen how to do this a little bit, kind of, in some way. So I don't know if you remember, but back in chapter 11, here we are going, this is weird, we're going back and looking at chapter 11 again. And back in chapter 11, we showed you this crazy base, sodium amide. Remember sodium amide? That's what we used for nucleophilic aromatic substitution through benzynes. Just insanely basic, 20 orders of magnitude more basic than hydroxide anion. So using this crazy base, sodium amide, we told you that you could deprotonate alkynes, terminal alkynes that have an H on them, in order to make these acetylide anions, these carbanions. You can't isolate those carbanions and put them in a flask. You have to use them as soon as you make them. And so the only thing they taught you about to do with those acetylide anions is they taught you to do SN2, because that's the only CC bond forming reaction that they taught you. Now you know electrophilic aromatic substitution. Now you know Grignard reagent formation. This kind of SN2 thing is not common. Probably the more useful thing that they would teach you how to do would be to add that. Whoops, let me use a red arrow. Right, it would have been more important to wait to show you that until you knew carbonyl chemistry, because it's more useful to add that to carbonyl groups. Right, that's a more useful process than SN2. But back then in chapter 11, all you knew is SN2, so that's what they taught you. Don't know any people who've ever done SN2 with acetylide anions. Now this chapter, we're going to teach you how to take powerful bases, some amazing bases here and deprotonate alkyl groups that are next to carbonyls. It's kind of like the acetylide anion idea. So we're going to take bases and we're going to use those to pull off one of the protons that we're not going to typically draw for you that are on the carbon atoms that are next to carbonyls. And that carbon atom that is next to a carbonyl we refer to as the alpha carbon. Just like alpha amino acids from, if you've learned about nomenclature for biochemistry and amino acids, or beta branched amino acids. They use the same alpha beta. Notice that the alpha carbon is not the carbonyl carbon. There's no proton on the carbonyl carbon here. We're talking about the neighbor to the carbonyl carbon. The carbon that is next to the carbonyl carbon. And you might say, I'm never going to get confused about that. Of course you will. It's easy to miss that little line that distinguishes the carbonyl carbon from its neighbor. So you need to be able to recognize which carbonyls have alpha carbons next to them and how many H's are attached. So we're going to teach you about some bases that can rip protons off of the alpha carbon next to carbonyl groups. And when you do that, you'll generate these carbanions. And those carbanions can be used for SN2 reactions if you want, just like we did with acetylide anions. So that shouldn't be a completely new concept. Sorry, I don't have a carb. I hate attacking R. I like attacking carbon atoms. But you could use that to displace bromides. You could use that to displace iodides. You could use that to do SN2 with tosylates. The important is this makes carbon-carbon bonds. And organic chemists like me, like my colleagues, we love making carbon-carbon bonds. That's the kinds of questions we put on our exams. If you look at my exams or other people's exams, we ask questions that are related to making carbon-carbon bonds. So that's how you make molecules bigger. Now, we could do the arrow pushing. We could draw this same mechanism using a slightly different set of arrow pushing. And I want to draw that out to make it clear that there's more than one way to correctly draw this arrow pushing mechanism. That if I take the same starting materials and the same base, there's a different way that I can push the arrows. I can also put, whoa, you know what? I'm realizing up above here that my arrows don't match the resonance structure. God, okay, I apologize for this. I'm going to have to erase these. I'll just scratch it out here. What I meant to do up above is show you this arrow pushing. That's the arrow pushing that's like the acetylide anion. And that blue arrow I just added is the one that gives you that resonance structure. I jumped the gun and drew this more common. The one I started with was supposed to be the second way to represent the same thing. So do you see that on top here, you can make the arrow, the blue arrow, I don't know if you can tell that that's blue, end directly on this alpha carbon in order to leave a carbanion. So, but there's another way to draw this arrow pushing mechanism down below, and that's pushing the electrons all the way to the carbonyl. And the book will tend to favor this. Most synthetic organic chemists, once you get out of sophomore organic chemistry, will favor this alternative depiction. It's the same mechanism, just a different way to depict it. And why do I like the mechanism above, even though they're equivalent and they're both worth the points? Because when you're first starting out, I feel like it's hard for you to see that the yellow carbon is the nucleophilic carbon when we draw it in this enolate form down below. So we would refer to both of these species here, no matter which resonance structure we draw. These are resonance structures. Those are called an enolate. And if an enol is nucleophilic on the yellow carbon, then an enolate is even more nucleophilic on that yellow carbon, on that alpha carbon. So nucleophilic. And it doesn't matter which resonance structure. These are the same mechanism. They're worth all the points. Doesn't matter which way you choose to draw this. Both are equivalent to each other. Just sometimes people like to draw the top mechanism depiction, and sometimes people like to draw the bottom mechanism depiction. I'll accept either one. Right? Enolates. They are nucleophilic on that yellow carbon. Powerful nucleophiles for carbon-carbon bond forming reactions. That's what I love. Carbon-carbon bond forming reactions. Okay, I'm going to stop there. And when we come back on Wednesday, we're going to be talking more about the specifics of which kinds of carbonyls are acidic, how acidic are they, and we have to decide very carefully what kinds of bases do we use depending on the carbonyl group? We're going to have to make some careful choices there in order to stay in alignment with the textbook. Happy Halloween. The best way to celebrate Halloween is to do organic chemistry problems to get ready for the exam while you're eating candy.